Question 6. Which statement explains the specificity of enzymes? Statement A states that an enzyme is the protein which acts as a key and fits the lock of its substrate. This statement is true. Enzymes are protein in nature, which means they are made of amino acid monomers linked by peptide bonds between the amino acids. This chain of amino acids is going to fold into a specific three-dimensional conformation which gives the enzyme or this protein molecule its specific shape. In this enzyme, there is a site known as the active site which is complementary to the shape of its substrate. This substrate is going to be able to sit in this active site due to the complementary shape. However, in the lock and key hypothesis that explains how an enzyme works, the enzyme is actually the lock while the substrate is a key. Just like how we bring the key to the lock, we bring the substrate to the enzyme, and the substrate is going to sit within the enzyme just like how the key is going to sit within the specific space in the lock. Therefore, it is not true to say that the enzyme acts as the key, because in this case it is the lock while the substrate is the key, and the second half of this statement is wrong. Statement B states that the active site of an enzyme only binds to a substrate at its optimum pH. This is true to a certain extent. The enzyme will have a specific 3D conformation, but this conformation will change due to the unfolding of this structure at pH levels that are far away or extreme pH levels, very different from its optimum pH. However, at pH level that is relatively close to this optimum pH, the active site can still allow the substrate to sit within itself. This statement, however, doesn't explain why enzymes are specific, because binding to a substrate at an optimum doesn't explain why the enzyme is specific to the substrate. It is specifically the shape of the active site that allows for the enzyme to be so specific to the substrate. Statement C states that the reaction between an active site and the substrate increases the activation energy. This is not true. An enzyme can help decrease the activation energy of a reaction, and this is how the enzyme helps to catalyze the reaction and make it faster. Statement D states that the shape of the substrate is complementary to the active site of its enzyme. This is true. Therefore, option D explains the specificity of enzymes, and it is due to the specific folding of the enzyme or the protein that allows the substrate to sit in this active site due to the complementary shape.